is open,
Yeah.
and the side door open, it provides enough light to the room because that
I just tried
washes.
turning it off though. And
It wouldn't turn off that one?
Well, the way to do it is to get the overhead lights on dimming the other two.
Yes, and turn that one off, yeah.
So that you turn them off.
Alright, so those lights are good, and then...
Yeah, I don't know.
There
Oh
we go.
yeah, great,
Yep.
perfect. Okay, thank you.
Perfect. Ha, much better.
Great.
Okay, so now go to full screen.
There we go.
I'm actually gonna try this.
Well, here, you can go. Do you have any particular reason to?
Let me just try to. Oh yeah, that works. Okay, great. All good.
Okay, I'm going to introduce you.
Thank you. Where do you want me to?
I don't have any embarrassing photographs of you either.
That's okay. Everyone's seen enough.
Yeah. Okay, everybody, let's get going. So, so I want to introduce Leah Werner, who is just defending her master's thesis here. This is really great stuff, as you'll see. So Leah came to me in an unusual way. So she came out of criminology, of all things. And it was like, what? What is she applying to a place like Scripps for? But it turned out that she had this really interesting, diverse background. So she'd worked on birds in South Africa, right?
Yeah.
And it kind of went on from there, this kind of long list of all sorts of things that didn't sound exactly like criminology to me. And then when she got to Scripps, we had this conversation. Well, what is your thesis going to be about? And she says, well, I'm really interested in management of fisheries. And of course, I'm a paleontologist, so it's like, well, okay, but we can look at the historical records of some of these fish groups, and we hit right away upon cod as the taxon of choice, because cod have been collapsed by human fishing in the Northeast. And elsewhere in the North Atlantic, and so this is a very interesting system to work on. And so we spent, I don't know, probably at least 6 or 8 months trying to make cod work, and that was a problem of finding appropriate sediment cores that had decent dating that we could collect a record, in this case of fish teeth or cod teeth, to try to reconstruct the historical record of cod, going back way before the time of the first fisheries data. And you'll see that that's the theme of her talk today, but not on cod. So we couldn't get cod to work. Part of the problem was that the sediment cores that we needed would all have to be in relatively shallow water, and just detecting fish teeth in a bunch of sand, particularly glacial sand from the North Atlantic, would be tough. So we switched to salmon, and salmon seemed like they were going to work. We had some nice cores. We got samples from those cores, and no salmon. Dang it. And so that was getting to be a little frustrating, because it's like, Leah, what are you going to do for your thesis? So then we set on this project that you'll hear about, which was to look at Southern Ocean fisheries. Notice the fisheries theme, okay, through all this stuff. And I have to really hand it to Leah, okay, that she stuck with her guns. This is what I want to know, is about these sorts of issues related to the management of fisheries. And so that is the underlying theme of everything that she's done. And I sure hope that she goes off and does something like that, post-master's degree. In fact, it turns out that people in Dalhousie, and I know Leah doesn't really want to go to Dalhousie, but anyway, they've been trying to recruit her to do the cod thing, of all things. With that, I'm going to turn it over to her. She's going to tell you all about fish teeth and the wonders of them. And I should also mention that this fits in very nicely, actually. It was sort of an outgrowth, to some extent, of work done by a former master's student, Michelle Zill, who's now a PhD student up at Riverside. And Michelle worked on a record that you'll see in a moment, which turns out to fit hand in glove, okay, with the data that Leah has produced. But Leah adds an extra little spice to the whole thing, which is really great. Anyway, so Leah Werner.
Thank you all for coming, and thank you, Dick. I am about to present to you my research from the last 2 years, and it's titled A Pleistocene Record of Global Fish Production and Implications for Sustainability of Polar Fisheries. So let's proceed. So my interest, as Dick pointed out, is in the international management of fisheries. I find it a really interesting resource to manage because of how fundamental of a commodity it is worldwide, especially in developing countries. It's also difficult to manage because it's interdisciplinary in nature. It has scientific, political, and socioeconomic implications. And currently we're struggling to maintain sustainability of many fisheries around the world. And this is especially true in the polar regions. So if you turn your attention to this figure, this shows the number of exploited and collapsed fishery stocks as a percentage of total stocks. So blue means that there's very low number of exploited and collapsed fisheries, and then all the way up to red is the most amount of exploited and collapsed fisheries. So as you can see, the Arctic in the northern hemisphere and then the Southern Ocean in the southern hemisphere have the most number of exploited and collapsed stocks. And so a lot of times fisheries collapse because we don't have a long-term data set to tell us about how these fishery stocks vary over natural climatic variability. So with that, I structured my study design around this dilemma. Is polar fish production high enough to sustain current industrial fishing? And by polar regions, we're referring to the Arctic and the Southern Ocean. But before we can answer that question, let's understand what the polar regions look like. So the Arctic and the Southern Ocean are different in many ways, but they also share some fundamental key features, such as low temperature, extreme seasonality, and the seasonal advancement and retreat of sea ice. So in the spring, when sea ice starts to melt, there's a bottom-up effect of biological production through the entire system. And this is because more light becomes available in the spring, and this warms the surface waters, and that inhibits any vertical mixing. So phytoplankton, which are a primary producer, and nutrients are stranded near the surface water. So this high light environment and this high nutrient environment is a really great recipe for phytoplankton to bloom, which we refer to as summer or spring blooms. They also they both are structured in that they have diatom-dominated food webs and efficient trophic transfers. So I'm gonna go over those two features a little more. So a trophic level is an organism's position in a food chain, the number being the positions away from the base. So in a nutrient-poor environment, what we refer to as an oligotrophic environment, you have a very tiny picoplankton at the base of the food chain. And so they're about less than a micron in size, give or take. So they are able to convert about 100% of the energy from the sun and to use for their own. However, as you go up the food chain, there is energy being lost at every trophic step. And the percentage at the bottom is about the percentage that you can convert, that organism can convert into their own tissue. So by the time you get all the way up to fish, all the way at the top, they only have a little amount of energy available to them. So these type of low nutrient environments are really not great for fish production. But let's return to the polar ecosystem. So these are dominated by diatoms. What's a diatom? Well, they're primary producers and they form the base of polar food webs. They are algae, and unlike other types of phytoplankton, they construct their cell walls with silicic acid. And they tend to be about 125 microns in size. So in comparison to that picoplankton, they're about 125 times that size, which is important because fish are able to predate directly on them, and in that trophic transfer, they're able to get a lot more energy. And so once Alfred Bigelow, who was an oceanographer, said that all fish are diatoms, in the sense that a diatom-based food chain will support a high level of fish production. Okay, so how did diatoms rise to ecological dominance in the polar regions, and how did that affect the evolution of modern fish production? So to answer this question, let's venture back 34 million years ago. So 34 million years ago, during the Eocene-Oligocene transition, the Antarctic circumpolar current formed, which increased upwelling of nutrients and biogenic silica production in the Southern Ocean. So this in turn, because diatoms construct their cell walls with silicic acid, this allowed diatoms to flourish, and they formed the base of many food webs. So by 15 million years ago, nutrient trapping in the circumpolar current built silica inventories that allowed the radiation of diatoms. So the diatom diversification in turn supported evolutionary radiations at upper trophic levels, such as Antarctic krill, whales, penguins, seals, and even fish. This picture on the top left is a picture of the modern ocean, and it looks at silicic acid concentrations in the surface water. So as you can see, near the poles, especially in the Southern Ocean, purple indicates low amounts of silicic acid, and then as you go up the color spectrum, that indicates higher amounts. So near the poles, they have higher amounts of silicic acid, which again is which is indicative of diatoms. So it seems that the polar ecosystems have evolved into productive regions, and this would in turn support fish production. Well, recent catch records from polar fisheries would suggest otherwise. So this left graph shows catch data in the Southern Ocean. So this is from 1960 to 2005, and this is in terms of thousands of tons of fish that were caught. So in the late 1960s, the fin fishing industry ventured into the Southern Ocean, and large-scale and unregulated fishing radiated from one stock and geographical area to the next. And so this triggered reductions of up to 90 to 95% of some stocks. A similar story unfolded in the North Atlantic. Oops, wrong way. So this graph is showing catch data from 1950 to 2000, but this is in terms of millions of tons of fish caught. And this is also only showing upper trophic level fish. So in the North Atlantic, some of the largest fisheries in the world were reduced in size, and they also were reduced by 90 to 95%. And these graphs are also indicative of catch effort, not only biomass alone, because there's clearly more fishing going on in the North Atlantic. But overall, they tell the same story, that there's these mass reductions of certain stocks in certain geographical areas of these oceans. So let's return to the underlying question that is supporting my research. Is polar fish production high enough to sustain current industrial fishing? Well, there's a paucity of data that is around to answer this question. Most data on fish production is based on fish landing records, and those typically only date back to the 1960s when industrial fishing began. So we're essentially studying a system that has already been exploited by humans. Also, in the polar regions, we are lacking primary and secondary production data. And this is because a lot of our primary production data comes from satellite images in order to estimate chlorophyll and use that to estimate primary production. But because the polar regions are dark for about half the year, we aren't able to get a high enough solar zenith angle to get our satellite images. So we are lacking primary and secondary production data in these regions. So how do we assess these long-term trends in fish production then? Well, we use ichthyoliths. So ichthyoliths are microfossil fish teeth and shark denticles. I actually took this picture from my own samples, so that is what they look like. So shark denticles are dermal scales. So ichthyoliths are found in almost all marine sediments. They reach the seafloor either through a live or dead specimen or through fecal matter. And they're composed they're very well preserved in sediment because they are composed of calcium phosphate, which is highly resistant to dissolution. So where other bones might dissolve, ichthyoliths have a better chance of lasting in the geologic record. They also capture mid and upper trophic levels. So unlike other biological proxies like nanofossils or foraminifera, those only capture the base of the food web, where because ichthyoliths are coming directly from mid and upper trophic levels, you can understand what's happening in terms of trophic efficiencies in these systems. So all in all, this is a great proxy for understanding what fish production looks like over time. So with that in mind, I'm going to structure my study design in support of my central question. So one of the questions I have that I would like to answer is, what is the baseline trend of polar fish production through recent geologic history? And by recent geologic history, I'm talking about in terms of the modern ocean configuration, but prior to human influence. And then with that, I would like to compare to an even earlier record and longer term record to understand how production has varied through time. So this is my study design. I am going to present to you an ichthyolith record through the Pleistocene. Pleistocene is an epoch that lasted from 12,000 years ago to 2.58 million years ago. My record lasts from 1.8 million years ago to the present. And so I use ichthyoliths to look at a proxy for fish production through this time. And I use 6 deep sea sediment cores ranging in latitude and oceanography as a context for what polar fish production looked like in comparison to other sites around the world. And then I have 67,000 year spacing between each of my samples in each of my cores. So then to answer the second question, I use an existing record. So this was a record that spans the Eocene-Oligocene transition. It is between 23 to about 42 million years ago, and this was produced by a former graduate student, Michelle Zill. She also looked at ichthyoliths as a proxy for fish production, and she used 7 deep sea sediment cores, also ranging in latitude and oceanography, and the spacing between each sample was about 139,000 years. So to put this timescale into perspective, before either of our timescales, at the way 66 million years ago, you have the KPG boundary, which was a mass extinction event. And this also includes this extinction of non-avian dinosaurs. But then you start to get within one of the records I'm looking at. Diatom-supported food chains started to occur when the ACC formed, and that was about 34 million years ago. So that was captured in my Eocene-Oligocene record. And then in between that, you have the diatom radiation 15 million years ago. And then within the tail end of the Pleistocene record, you have Homo sapiens emerge about 300,000 years ago. So I hypothesized that based on all of the reasons we discussed, that the higher ichthyolith accumulation will occur at the poles relative to lower latitudes. And again, this is because there's short diatom-based food webs, and all fish are diatoms, according to Alfred Bigelow. So let's go into where our sites are located. So all of the sites you see in yellow are in the Pleistocene record. Those are the ones I produced that spanned the last 1.8 million years. And then the green ones are from the Eocene-Oligocene transition. And these sediment records came from the Ocean Drilling Program, which is ODP, or the International Ocean Discovery Program, IODP. And then from the Eocene-Oligocene, you also have a Deep Sea Drilling Program record. So within the Pleistocene, I have 2 high latitude records, one in the subpolar North Atlantic and one in the South Atlantic in the Southern Ocean. And then I compared those 2 sites to lower latitude sites. 2 that are gyromargin sites, one in the South Pacific and one in the North Atlantic, and then 2 equatorial sites, one in the Atlantic and one in the Pacific. And collectively, in between the gyromargin and the equatorial sites, I'm going to be referring to them as low latitude sites. And then within the Eocene-Oligocene record, we have 2 Southern Ocean sites, as you can see right here. We have North Pacific, an equatorial Pacific, and a South Pacific site, and a North Atlantic and a South Atlantic site. And collectively, those will be referred to as low latitude sites. So how did I process my samples? Because all of these came from sediment cores. So my collaborators at the Ocean Drilling Program drilled into the open ocean to retrieve these sediment cores. And then when I ordered them, this is what they looked like. That is my sediment sample, essentially. So I put those in the oven, and then I got a dry mass weight that I will use in my ichthyolith accumulation rate. And then from there, part of the point of processing is to remove all elements that are not ichthyolous, so it's easier to visually identify them. And so one of those components that I wanted to get rid of is carbonate. And so I used acetic acid, about 10% of it, and I dissolved my samples in them. So any carbonate would react, as you can see. And when it finished reacting, which would take about any 2 to 24 hours, I washed it in a 63 micron sieve, and then I put it in the oven. Lots of oven time. There's another component I wanted to remove, which was silica. So we used heavy liquids, and when diluted, it will allow silica to float to the top because it's lighter in density, and ichthyolous to remain at the bottom. So I was able to just pour off all of the lighter fraction and just keep the denser fraction, which included ichthyolous, intact. So, and then went back in the oven. So I had to do this about 160 times. I did have a little bit of help with an undergrad. There's an additional step I had, which was for one of my sites, my North Atlantic site, there was still a heavy amount of residual sediment left. So I used alizarin red S to dye it, and it dyes anything with calcium phosphate. So it's easier to visually identify all of the ichthyolous when under the microscope. So now we have all of our samples processed. It's time to get to work. So I picked and counted every ichthyolith and throughout all of my sites. So once I have my raw number of ichthyoliths within each of my sediments, within each of my cores, how does that translate to proxy for fish production? Well, this is ichthyolith accumulation rates. So this takes into consideration the number of ichthyoliths within each sample and multiplied by the dry bulk density of the sediment and sedimentation rates. So this is a metric of ichthyolith flux to the seafloor in a given area over a given time. So an increase in IAR is translated to an increase in ichthyolith production, which in turn is an increase in fish production. It's a proxy for that. So it normalizes for any changes in sedimentation and lithology, so you're able to compare a site that's in red clay to a site that has a pelagic nanofossil ooze. And that's because it's incorporating the sedimentation rates into that calculation. So I used all records that were correlated to astronomically tuned chronology, so I understood what one of my samples, the age of it, was at any given site. So now that we have all the data processed and all the samples processed, let's return and see what the results are. So again, one of my questions was, what is the baseline trend of polar fish production through recent geologic history? And I expected that the higher latitudes would have higher ichthyolith accumulation relative to lower latitude sites. Well, it turns out I was very wrong. So let's take a look. So this is my Pleistocene record. Again, this spans the last 1.8 million years. And each site represents each line represents a different site. And it shows the temporal variation in each of those records. So the x-axis shows time, and then the y-axis is IAR. And for reference, the colors are coordinated to the map on the far right, and you can tell where each of those came from. So you can see temporal trends through our study interval. However, our main focus was to compare baseline trends and how they varied between each site. So keep that in mind. So IR was found to be statistically comparable between polar sites, but not between polar sites to low latitude sites. IR was generally 1/10 of lower latitude sites. And so we can see that stark difference just in the mean values alone. So you can see these are my two high latitude sites, and they have generally very low variability, and they are 1/10 lower than all of these low latitude sites combined. And you can see the stark difference even in the fact that most of the minimums of my low latitude sites are still higher than the maximums of my high latitude sites. So all in all, the main takeaway from this is that IAR at the high latitudes is an order of magnitude lower than low latitudes. Again, this was not what I expected at all. So you can see this in a different way. You can see this is, again, my Pleistocene record, And each of these box plots represents the spread of IAR at one of my site locations. So, and latitude is shown on the x-axis, so you could see where all of these sites are located. So 704 is my Southern Ocean site, and 982 is my North Atlantic subpolar site. And then these are my low latitude sites collectively. And you can see there's a clear latitudinal trend in ichthyol accumulation over this time interval. So now that we know that I was completely wrong and the Pleistocene was a lot more favorable for high fish production in the lower latitudes than the high latitudes, let's see if this trend holds up in the Eocene Oligocene. Well, it's the same exact trend. So low IAR is evident in the high latitudes since the late Eocene over this time interval. So this is the same graph you looked at, except now I have added the 7 sites within the Eocene and Oligocene. And you could see our 2 Southern Ocean sites, 689 and 748. And they are much lower than the other lower latitudes. They are about 1/10. And there's one exception to this trend, and that is that the gyre center sites within the Eocene-Oligocene transition are also statistically similar to the high latitude sites. And the explanation we can think of for why this would be is because both in the modern ocean, gyre centers are ultra-oligotrophic. And again, oligotrophy is indicative of low amount of nutrients and it's not really great for fish production. And these gyre center sites are 886 and 596. So you can see those are also both very low. But generally, you're, again, seeing the same exact trend. IAR at the high latitudes is 1/10 to that of the low latitudes. And so this is all of the samples that throughout both of the records put onto one graph. So you can see my Pleistocene record is on the left and the Eocene-Oligocene record is on the right. And each dot represents a separate sample in time. And it is placed on, and each value is indicative of log 10 IAR. The colors are indicative of proximity to the equator. So red is closer to the equator and yellow is closer to the poles. So again, there's this clear latitudinal trend that high IAR is evident in the lower latitudes and low IAR is evident in the high latitudes. And there's 2 trends I'd also like to point out. I have several zeros in my Pleistocene record, and those are all within my high latitude records. So I just didn't have any ichthyoliths within some of these samples, which was not the case in the Eocene-Oligocene record. All of them had at least some amount of ichthyoliths. But another trend is that my low latitude sites generally have higher IAR than the low latitude sites in the Eocene-Oligocene. And one reason for that might be just different locations. I had some of my samples in gyre margin sites in equatorial regions, which are generally better for fish production. Whereas in the Eocene-Oligocene, they have gyre center sites, which again are more oligotrophic in nature. So the main takeaway from all of this is that the high latitudes have seemed to have consistently low IAR, which is a proxy for fish production. And this is evident in both the Pleistocene record and the Eocene-Oligocene record. So despite the rise of diatoms to ecological dominance and the diversification of upper trophic levels, we're not seeing this translated to a high level of fish production in these polar regions. So what accounts for this? Well, we aren't sure, but we have looked into 6 possible explanations for that, which I will go over individually. So first, in the polar regions, extreme seasonality is a prominent feature. There are strong seasonal changes in solar radiation, in ice cover, and in nutrients. So you can see this clear seasonal trend in the satellite image of the Southern Ocean, which you can see on the right. The colors are indicative of biological production. So you have purple, which means low primary production, and then as you get more into the yellows and red, that means higher. So in the spring, you can see the spring bloom, and same with the summer, less so in the fall, and then very minimal in the winter. And you can also see that in the winter, you have a lot more black, which means that there is actually no satellite imagery of this at all. And again, that's because this area is in almost complete darkness during the winter, so we aren't able to get satellite images. So you have a pulse in primary production in the spring, but then for the winter, there's very minimal. So there's a very narrow window for fish to reproduce and grow. So that forces them either to reduce their metabolic rates or to migrate to lower latitudes where they'd find more food. And we see an example in the modern ocean of why some fish are unable to live near the poles, and this because of photoperiod. So for mesopelagic fish, they undertake diel migration, which is vertical migration through the water column. When it's light out, they descend to deeper waters to avoid predation, and then when it's dark out, they'll ascend to predate. And so because of this extreme seasonality that changes photoperiod, you have almost complete light during the summer and then almost complete darkness during the winter. This is really not a great recipe for this diel migration. So these mesopelagic fish tend not to live in these high latitudes. So all in all, the seasonality and resource constraints and energetic commands added with the cold temperature is an explanation for why we're seeing this low IAR trend in these high latitudes. So another explanation is phenology. So everything in polar regions is dependent on seasonality and is variable in time and space. So predators will often time their reproduction based on the seasonality of their prey, so that they can provide maximum food supply for their larvae. So this trend is represented in this diagram right here. So this diagram shows phenology, which is timing, and then abundance in terms of biomass, and the effective degree trophic match or mismatch. So the key variable is the degree of trophic overlap, which is shown in the gray area. And so the trophic overlap shows predator needs in the continuous line and then prey availability in the dotted line. And then the bell curves are indicative of when they're most available in terms of biomass, and it's placed on the x-axis of time to show when their seasonality. So let's use the example of the polar regions. Phytoplankton bloom in the spring, so this would be the dotted line, they're the prey. And this is due to the seasonal retreat of sea ice. And then fish, which will be the continuous line, they time their reproduction right after this so that their larvae can get the most amount of food available to them. So this is a trophic match. However, the seasonal retreat of sea ice can change based on temperature. So warming waters can make phytoplankton blooms occur earlier in the year. So as you can see in this case, phytoplankton are blooming earlier in the year because the sea ice has melted earlier. But fish never got that memo, so they're going to reproduce at the time that they think that this spring bloom will occur, but they're going to miss it. So this will be an unfavorable condition for recruitment. And so over time, this will suppress fish production in these regions. So that was a direct influence on fish production for those that directly predate on phytoplankton. But this can also cascade up food webs for higher trophic level fish. So both polar ecosystems are, in certain areas, have wasp waste ecosystems. And this is when a small number of intermediate level species dominate energy flow from the lower and upper trophic levels. So in the wasp waste ecosystem, that term comes from the idea that intermediate trophic level species form the narrow waist through which energy flows in that ecosystem. So let's use Southern Ocean as an example. So this is a food web of the Southern Ocean. Krill are that intermediate level species. So you can see krill in the middle. They provide food source for so many apex predators, including fish. And so any variation in their populations will have a direct influence on the predators relying on that, including fish. And there's not a great alternative in terms of a food source for krill. Krill are pretty much all there is in certain regions of the Southern Ocean. And so fish rely exclusively on krill in some areas, so any variation in their production in time and space will also have an effect on fish production and results in low IAR seen in our record in these regions. So another explanation is the glacial interglacial cycles. Now these have occurred over the last 2.6 million years, and they occur because of the variation of the Earth's tilt that causes a different amount of solar radiation to be let in. So polar regions are disproportionately affected by these cycles because they already have less solar radiation input than other regions of the globe. So this results in changes in size and location of continental ice sheets. So the glacial period is what we refer to as an ice age, and it's characterized by glacial advances in extremely low temperatures. And you can see the last glacial maximum. This is the northern hemisphere. You can see that it's almost completely covered in continental ice. And then this is today's day, and we're currently in an interglacial cycle, and that is characterized by rising sea levels and a reduction of sea ice. So this occurred throughout my Pleistocene record, throughout the last 1.8 million years, and the cycle occurs about every 100,000 years. So you have this flip-flop between ice age and warming climate. So this, of course, is going to have effect on fish that live in these Arctic regions. So when it was completely covered in ice, fish were either migrated to deeper waters or they migrated to lower latitudes. And then when the next interglacial period came, they would recolonize in the Arctic. So you have these repeated community shifts where either fish are forced to go extinct or any fish with a niche overlap will be adversely affected by this. And so overall, this will decrease and suppress their production over time. So this played out slightly differently in the Southern Ocean. So this is a picture of the Southern Hemisphere. And you could see Antarctica in the middle. This is, again, on the left, you'll see the last glacial maxima. And then today we're in the interglacial cycle. So fish, during the first glacial maxima, they may have migrated out of the Southern Ocean to lower latitudes. But because of the strength of the ACC, that's the Antarctic Circumpolar Current that traverses the Southern Ocean, and that also acts as an effective biological barrier, fish were unable to return back to the Antarctic continent in the next interglacial cycle. So either they migrated out and stayed out, or they stayed near the Antarctic continent throughout all of these cycles. So that's why you also see these local adaptations close to the Antarctic continent, where you won't find elsewhere in the world. So all in all, these cycles had a profound effect on fish fauna during this time interval. So we just talked about how fish weren't able to generally leave the Southern Ocean, and they persisted during glacial periods. But other competitors that shared a similar prey resource were able to traverse that ACC, and that includes whales. So giganticism in whales rose about 3 million years ago, and that's a direct reaction to the shift between homogeneous to heterogeneous upwelling near the equatorial regions that caused whales to migrate to exploit patchy prey. So by adapting to a larger body size, they were able to expend less energy on these long migrations. So it's a selective advantage. So all in all, they were able to effectively exploit krill, for instance, and fish who also predated on krill weren't able to effectively exploit these patchy populations of krill. So over time, fish may have evolved into a less dominating role in the trophic web of the Southern Ocean, whereas whales might dominate. And so in today's modern ocean, most carbon flows to the diatom krill apex predator food chain, and the trophic role of fish is largely overshadowed. So this might have short-circuited the trophic role of fish over time, which is why we don't see as high of production in these regions. So our last explanation of why we're seeing this low production trend for fish in the high latitudes is because of life history strategy. So life history is a theory in evolution that explains aspects of organisms' behavior and anatomy. It also explains how natural selection has selected for certain traits to maximize evolutionary fitness or reproductive success. So because the polar regions have short-term and long-term climatic variability, many fish have evolved to have K-selected life history strategies, and this includes delayed maturity, slow growth, large size, short spawning durations, and longevity. So let's focus on a couple reasons why that is, because these fish are adapting these life history traits as a reaction to their environment. So for slow growth, for instance, there's a lack of biological production throughout the winter, so they're left without a lot of food, and so they have to reduce their metabolism. And adding to the fact that there's already really cold temperatures, they have pretty low metabolism throughout the year. So this results in slow growth. Also, longevity is a selective advantage in these regions. Because you have this extreme variability, fish that can have many year classes can survive long periods of unfavorable recruitment conditions, whereas fish with only a few cohorts might collapse before a favorable recruitment condition returns. So longevity would allow fish to, over time, to persist, whereas any short-lived species would not fare as well. So we can see examples of this longevity in some of the largest fisheries in the world, and those are within the Southern and North Atlantic Oceans. So orange roughy is in the Southern Ocean. They live over a century. Atlantic cod, which I'm sure a lot of people in this room have eaten, they live to 25 years, and they're found in the North Atlantic. And we have Antarctic toothfish, which live about 35 years. So fisheries with these traits will inherently support fish populations with fewer individuals and a greater sensitivity to climatic change. So let's return to the 6 reasons underlying why I think there is low IAR, which is again a proxy for fish production in these high latitude regions. So they all sum up to one explanation, which is that it's hard out there for a polar fish. So in sum, I produced a Pleistocene record that spanned the last 1.8 million years, and I used ichthyolith production as a proxy for this fish production. And then I compared these results to an existing record through the Eocene and Oligocene that spanned the last 23 to 42 million years. We were ultimately looking for an explanation of when the modern polar ecosystems evolved in terms of fish production, and we did not find that answer because our results point to the fact that the high latitudes are an order of magnitude lower in IAR than the low latitude sites. So the evolution of polar ecosystems that facilitated the rise of diatoms, efficient trophic transfers, and the diversification of upper trophic levels did not translate to enhanced IAR. So indeed, there appears to be intrinsic limitations to primary and secondary production, which in turn would affect polar fish production in these regions. So returning to the foundational question of my research, is polar fish production high enough to sustain current industrial fishing? Well, my results suggest otherwise. We may be mining polar regions of fisheries with intrinsically low populations, as shown in my records. We don't know exactly how IAR translates to stock sizes, but we assume that low IAR means a lower rate of stock production. We also know that these 6 explanations of why we're seeing this low IAR in these high latitude regions also suggest that these are regions we probably do not want to be overfishing. So in sum, these polar fisheries have fewer individuals, a greater sensitivity to perturbation, and a greater proclivity for collapse. This is also exacerbated by climate change and overfishing. So warming temperatures have an inverse relationship for successful fish recruitment. So even though fish in these regions have persisted in the past during other warming events, this one is the modern times is a different story because not only are we experiencing warmer temperatures, but we are also experiencing overfishing. So we're overfishing some of these populations at a rate that they are unable to naturally replenish. So we may, in fact, be driving these polar stocks to the brink of extinction. And with that, I would like to thank everyone here. Thanks. But also, I would like to thank my committee members, Dick Norris, Dave Checkley, and Phil Hastings, and then also everyone within my own lab. They've been so foundational for in supporting me through my research. Also, I'd like to thank several professors I've TA'd for, because that has given me money. But also, they've been great guides for teaching as well, especially Jane Tran is who I TA'd for 3 quarters. And then some others, Jessie Koo, she helped me with processing some of my samples. Elizabeth Siebert, who went through Dick Norris's lab and also provided me with some insight of my research. And then Michelle Zill for providing me with my Eocene Oligocene record. And then we also have my core sources. And also, thank you for everyone pictured in here and everyone also not pictured in here. I don't have pictures of everyone. But overall, everyone, my family and friends have been hugely helpful during this time for my research, and I really want to thank everyone that I have included in these slides and those who I haven't. So, thank you. I'm going to go back so this isn't distracting. Is there any questions? Yeah.
Okay, so I recognize that this isn't part of your record, but
Sure.
I thought it was surprising that at least in one of the mid-latitude sites in the Eocene, that there was a pretty high IAR.
Yeah.
Considering that there's not any proliferation of diatoms at that time, right?
That is very interesting. And I think Michelle Zill did go into that in her thesis as well.
Do you, what was maybe her reasoning of, because I just would have thought for sure that it would have been lower considering how important diatoms are.
Let me go to that slide so everyone knows what you're talking about. So we actually expected, I know that Michelle expected to find higher IAR in a lot of her locations, but are you referring to 522?
That, particularly in the Eocene, right? Because that would have been before
diatom.
Yeah, right.
Yeah. No, I don't remember her explanation. I don't have a great explanation for that either.
I mean, in one sense, it maybe could, I guess, kind of be like an outlier, right? Because you can see the rest of the sites, it is low like you might expect, right?
Yeah, and like I said before, some of these are within oligotrophic areas. In this one, I don't remember the exact location of where this one is. We can look at our record.
South Atlantic.
South Atlantic.
There's lots of slides, huh?
Yeah.
So that would be 522, yeah, right there. So I don't have a great explanation for why we're seeing that high trend. Dick, would you be able to provide some insight into that?
Not really. I mean, I guess that it's striking what your statistics had showed that overall the low latitudes in the Eocene and Oligocene apparently have a lower IAR than the Pleistocene records. And you know, we sort of have a facile explanation that maybe that's just because the site locations control this, but it could also be that coming back to this issue, you know, that overall production is less because the system hasn't flipped into a dominated state. But yeah, 522 is a confusing place because it's in the gyre. You'd think that it would be relatively oligotrophic, but that particular area gets a current that spins off the southern part of Africa and into the tropical Atlantic, and so it's possible that it's actually getting a nutrient source coming off of Southwest Africa. But that's a guess.
Sure. Cool. Great talk, by the way.
Thank you. You have any other questions? Cool. Makes it easier. Well, if anyone does have any questions, you can also ask me later. Yes, Dave?
What's the current status of fishing in the Southern Ocean?
So it has decreased from the state it was in the 60s and 70s. there's not as great of an effort, but there is also krill being more exploited. So I don't know the timeline of when the krill fishery started, but I believe it is later than the fin fishing industry, within the Southern Ocean at least. So I think that they have maybe started overexploiting krill, and because there's all of these, there's a prevalence of research on this like regime shift, resulting in these stocks being collapsed in the 60s and 70s within this area. And also there's a regulatory body now. There's definitely less fishing than there once was several decades ago.
Wasn't there an exclusion zone established just
There-
recently for the Ross Sea? Or?
I believe so. I'm not quite sure exactly where it was established, but I know that in the 80s, and I'm forgetting the name of the regulatory body, I should know that, but that was when it formed in terms of regulating fisheries management within the Southern Ocean.
Do you know much about the orange roughy fishery in that respect? Because I've always been impressed by orange roughy, very long-lived individuals, sort of the age of your great-grandparents or something like that, for one fish.
Yeah,
And I know that the New Zealanders closed off some of their
they did.
seamount-related fisheries, right?
Yes.
Any insights on that?
So I'm not quite sure in terms of recent research on how that has, because they have closed off some fishing areas to allow orange roughy populations to come back. But so I'm not quite sure where the status of those are now. I think it is also hard to tell since they are so long-lived. It would take a little bit of time to understand if that is helping their populations grow back. But yeah, I'm not quite sure about the actual stock status of them now. But I do know that they are quite expensive in Japan. So they're still on the market in some places.
I think they might still be on the market in bonds.
Wait, really?
I think so. Yeah.
I've never seen them in the.
Our fish, we sell these things in the U.S.
Yeah.
How long do they live again?
They live over a century.
That is amazing.
I know.
Wow,
I did not know that.
Are we eating something like that?
Yeah.
Yeah, what?
I know, right? All of these very large fisheries are very long-lived and slow-growing, so.
The one that really gets me is, it's unrelated to your thesis, but is the Arctica. It's a clam that's harvested off the U.S. East Coast. Those things are like 200 or 300 years old.
Huh.
And they turn them into clam chowder.
Oh.
It's like,
That is such a waste.
clam chowder, by the way.
Oh.
Clams. Yeah.
There are a lot of them?
What's that?
There are a lot of clams?
Well, yeah, they fished a lot of them. Of course,
Oh, yeah.
basically non-reproducing fishery.
So that's another key point is that even though some of the biomass might be high, production is different in the fact that well, at least my findings have shown very low rates of production. So that means that you can fish what seems to be an abundant resource, but it's not going to replace itself very readily.
Okay, any other questions? All right, well.
Thank you all for coming.
So I don't know what the plan is for parties.
Oh, lots of beer.
So, lots of beer. Is there
And Jell-O
is there
shots.
a plan?
Yeah, I'm gonna go over and open up Surfside. We have it all reserved. So I think
11:30 to 4.
11. Thank you.
Straight into TG.
Yeah.
People can meet there after.
Okay,
So-
we're gonna chase everybody out at this point, and the committee will stick around and clearly, be opposed.
Meet everyone.
You guys can hand.
What a cute little
Handy.
flag.
Handy guy.
Where should we go?
High five.
Thank you so
That was
much.
excellent. We'll see you in a bit.
Oh, well.
Oh, sorry.
You're doing pretty well.
Great.
And that he was the lost
I thought it was
first line.
supposed to be to the office.
I'm sorry, I meant to introduce you. Did it
Sorry,
out.
I didn't have anything to put in it at the time, and then
Usually most of those are need to be one.
Please. With all the ones that I, this is what I'm
So it kind of.
It's kind of indicative of me as a person, honestly.
Oh,
Dick, will you sign this?
I can sign. Do you have a?
Yes.
Masters, it's easier to chase down three.
It is, it's easier.
That's
right.
So it's a current that, sorry, around like all 7 ocean basins. So it's a way for nutrients to find so in the northern hemisphere.
Thank you very much.
No teaching this quarter.
Oh, yeah, so now I see like a lot of.
Yeah.
Thank you very much.
You've heard of that, that create a lot of storms in the western boundary basins, such as Japan, try to get more
Yeah.
tsunamis there.
Just set up
And then on the east coast, we have less, so that's why.
Rosenblatt partnership. It's gotta be in here.
So it's the entire current form. Yeah.
Huge drivers.
Did that do it?
I can show you.
Less than 3, but I guess I should learn.
Okay, thank you.
It's worthwhile coming in and learning the ropes.
Yeah.
Who's the Rosenblatt for sure? I forget.
We just announced the state of Japan.
Small.
Should I can grab a paper and just probably.
Minor celebrity.
I don't know why.
Yeah.
I guess the backlight's supposed to be a
I
better.
don't like these lights.
So I probably shouldn't turn the spotlights on.
All right.
Yeah, they're in the back.
I'll get it, Dick.